Hi everyone, welcome to the Pharmaceutical Clinician Hub, a platform aimed at closing the gap between clinical medicine and the pharmaceutical industry. I'm very excited and honored to have our guest speaker here today, Miguel Pereira, who has a medical background as well as a pharmaceutical industry background. He has worked at all the best institutions in Lisbon and the US, and he's now here in Cambridge. And he's going to talk to us today about drug and clinical development from the pharmaceutical perspective. Miguel, a pleasure to have you here today. How are you doing?
Thank you, Neil. All good, all good. And excited
Perfect.
to share a little bit about what people are doing in pharma to fellow doctors. Because it's quite a different perspective.
Okay, over to you.
All right, thank you. So the purpose of this is to share a little bit about what happens on the pharma side, because I do realize that before I was in pharma, I saw things quite differently from what pharmaceutical companies and also governments and payers see drug development. And in reality, we can see that there are three different interested parties in drug development. So on one side, we have the sponsor.
One real-time interruption. Are you able to just maximize from your side, Miguel? It's all good.
Oh,
Uh,
is it so I see what you mean. Let me get this. Because it shared on my other screen. So let me go back. How about now?
Perfect.
Excellent. Okay, cool. So essentially there are three interested parties when it comes to uh, developing and bringing new drugs into the market. So on one side, obviously we have the clinicians, so we all want to treat our patients and we want to make sure that all the things we give to them are safe, but at the same time are having a positive impact. They have some sort of efficacy. We have the pharma side, which is basically says, well, we want to do it fast. We want to get our drugs approved because we want to get some return on our investment. And that is essentially their incentives. They want to make money out of creating new drugs. And the third party, which is the one we're definitely not going to talk a lot about today, is the payers. So the payers could be either governments, so entities like the NHS, could be insurance companies, could be private people that want to pay for drugs, even though that happens rarely, and they would need to see whether there's value in whether people can access that. And if there is no good cost effective relationship between a drug and the impact it has, they will decide not to pay for it, and all the work that's been done into developing that drug is not going to be worthwhile. But yeah, that's the part we're probably not going to touch much aside from this first slide. So what we're going to do is going to quickly go through a little bit about the preclinical space and then move to the clinical space and the main three phases of clinical trials, phase one, two, and three, and talk about some interesting aspects of that I think we sometimes forget in terms of the interests of what a pharmaceutical company needs to see and what clinicians want to see. But first, just wanted to give an overview of all the work that comes into developing a drug, even before it reaches the first patient in the first phase one trial. And what happens effectively is that for each one drug, and you can see here in the IND enabling part, for each one drug that eventually might reach a phase one trial, usually there have been 10,000 plus drugs going from target validation. So a lot of this is actually basic science research still, going to hit discovery, lead optimization, until you get some candidates, and out of those you will choose one. In the process for that, also needs to clear certain gates. So you need to see if the drug is potent enough, and effectively, even at the preclinical level, you need to sort of make a very educated guess of whether that drug is going to have a good relationship between safety and efficacy in human beings. And for that, you have multiple models like PK, ADME. I've actually previously worked on building a model that predicts this sort of toxicity just based on chemical structure. So there's a lot of work done in that area. And then you need to work on standardizing your processes so that you have this. This is what is called GLP talks, which I'll talk a little bit about in a second, which is good laboratory practice in CMC, which this has to do with the manufacturing and having the processes for manufacturing at scale the drugs. So you need to have all of that into place even before you get into the clinic. And for that, the way we usually divide things in pharma, which perhaps some of the people watching this will have seen, is we have the GXP practices in pharma, of which the three main ones we're going to be touching on are the preclinical, which is we just talked about, which is the GLP, good laboratory practice. And then we have the GCP, good clinical practice, which is that it's mainly dedicated to clinical trials, going from informed consent to every single type of procedure that occurs in clinical trials. And then we have the manufacturing. And even though manufacturing is put here in the middle, I do think it is part of all this process, because even before you get to the clinic stage, you need to have already manufacturing processes in place, and you need to be able to produce your drug to a certain standard that will be allowed to be administered in clinical trials. And effectively, whatever procedure you develop now is what's going to be then used when the drug is produced at scale, at large scale for commercialization. And then, of course, there's GDP and GVP, but those are way after a drug has shown that it actually works. And an interesting thing, which I know is a very small image, and the point is that people are not actually reading this in detail. Um, and I just wanted to illustrate that when we think about this, the GMP side of things, so the GLP side, where we were talking about all those gates, when we go to the GMP side, so after we've defined our drug, we have something that works. There are a lot of processes put into place. I'm not an expert on this. This is just something I wanted to illustrate. This is an example for a process to manufacture mesenchymal stem cells. So that's what the MSC stands for. It's an extremely complex process, and everything needs to be well, partly because it's highly regulated, everything needs to be completely standardized, completely clear, such that the same procedure can be followed by a lab in the UK, a lab in Germany, a lab in China, or a lab in Peru. So in all those places, all the processes need to be well documented so that every lab could be able to manufacture this product to a high standard before it gets into a human being. So yeah, so this was just a quick passing through the preclinical process, where I also wanted to emphasize that the preclinical stage, where you go from tens of thousands of potential targets in potential druggable targets in molecules that will potentially become drugs, in terms of budget for a pharmaceutical company, that interestingly only counts to roughly 20% of their budgets. In some cases, going to 30%. But for most large pharmaceutical companies, that accounts for 20 to 30% of their budgets. In reality, if you could split the pie almost 20 to 30% preclinical, going to 30, 40% clinical trials, so clinical development per se, and the rest into post-commercialization or post-approval commercialization, real-world evidence, phase 4 trials, and marketing. And actually, marketing takes a fairly large chunk of this last piece of the pie. But yeah, so in terms of clinical trials, I would like to start with a question, and I'll be slow here so that people can think about what this means. So in a clinical trial, what question would you like to answer? And this pertains a lot to what I do at the moment. I design clinical trials as a statistician, and one of the things that I do is a special kind of trial, and hopefully this will help illustrate that. So in a trial, would you like to see the probability that treatment A is superior to treatment B? So treatment B could be a placebo. Do you want to see if the treatment A has a 90% chance of being better than the placebo? Or would you like to know what is the probability of seeing your trial data, so the data you end up collecting in your trial, assuming that the treatments are equal. So what would you like to know? What is the probability of seeing your trial data, assuming that, for example, your treatment and your placebo are the same? So if you think about this in a little bit, and you have one particular question, or you prefer, what I can tell is if you chose the first one, or sorry, if you chose the second one, you decided to do a classical trial. So in most clinical trials, the questions we are asking when we're comparing two treatments or a treatment against a placebo is what is the probability of obtaining certain data, assuming that the treatments are equal? On the other hand, if you want to know the first question, which is the probability that one treatment is superior to another, you actually want to do a Bayesian trial. Uh, and I'm just putting this out here because what my main expertise is Bayesian trials, and the reason for that is because from a clinical perspective, I believe it answers better questions. Um, I'm not going to give a lecture on Bayesian trials. This is just to illustrate it. And
That's the next time.
and exactly, that's the next one. That's a follow-up. Um, and the other thing I wanted to say is if you want to see the second question, and you want to know this probability of obtaining certain data, assuming that the treatments are equal, actually, you want to run a classical trial, and this probability that you get here is the p-value. However, this also shows, when you think about it this way, I hope this also shows that the p-value is just a probability that we see data that shows that the two treatments are different when we were assuming that they were not different. And therefore, we want this probability to be low. And usually if this probability is below 5%, we say that our trial was successful because there is a very low chance that we see data so showing the two things that are so different if I was assuming that they were the same. And this is also why some people might have seen that the p-value does not suggest the strength of association. So you can have very low probability. It does not mean that the treatment has a stronger association or not. On the other hand, if you look at things this way, the probability that treatment A is superior to treatment B, in fact, you want this probability to be very high, say 90%, 99% for overwhelming efficacy, and it will tell you how strongly that association is. But yeah, so the point that I want to make is, especially if you are more interested in the first question, most trials are actually asking the second question, which is the way the industry deals with that. The other aspect I wanted to share, which comes a little bit from my own clinical experience, is the gap between patient and data. If you are running trials, or if you are a physician, not necessarily running trials, just seeing patients as normal, this is what you see. You see a patient in bed suffering a little bit, or doesn't look it's his best, this gentleman. And for me, and I've seen this. However, now I don't see this anymore. What I do see is this. I see a bunch of data. I see a spreadsheet with doses that were given to patients, their patient IDs. I see the SOC, so the system organ class, in the preferred term, standardized preferred terms of what they have. And I see some form of grades for the severity of their adverse events. And what is interesting, at least for me, is working in pharma now, is this does not necessarily translate this. And effectively, I can look at these numbers and imagine that I'm seeing someone like this, or imagine that I'm seeing someone like this. And it's very difficult for us to actually capture what clinicians see in practice. And I think people that are involved in clinical care can appreciate that seeing a patient is very different from being told their medical history or their current history, and even less so when you have this in a standardized spreadsheet. So this is something that in pharma we still have not fully been able to capture. We might not, we're not sure if we ever will precisely. But yeah, so aside from this gap from between patient and data, so let's now navigate a little bit into the three phases of clinical trials. So for phase one, the main goal, there are two main goals, I would say. So one is to prove that the drug is safe. It's the first time that it's being given to people. And we want to know, can we give it to people or not? Is there a safe dose for this? And that's why this is formulated as getting the dose right. Effectively, one thing that I am always very skeptical about is the designs that are used for these phase one trials, and this is actually an area that I work on a lot. One of the main designs still is, even though it's being less used, is this 3+3 design, where it's extremely easy. It's what I say. This could be an elevator pitch about the 3+3 design. As we're going up an elevator, I can tell you how it works. So you start with your lowest dose in your trial. You add 3 patients in your cohort. If you don't see any DLTs, meaning dose limiting toxicities, you escalate to the next dose level. If you see one DLT out of 3, you add 3 more, which is why it's called 3+3. If you don't see any more DLTs, so you have 1 out of 6, you escalate to the next dose level. If you do see at least one more DLT in the new cohort that was just enrolled, you stop. You consider that the previous dose level was the MTD, and MTD means maximum tolerated dose. If you see 2 or 3 DLTs out of 3 patients, you immediately stop. If that's your first dose, you say that you didn't find a safe dose. Ideally, what happens is you go up to your 5th or 6th dose level, and then you determine that that is your highest safe dose. However, I mean, on one hand, this is an extremely simple design that someone would need practically no training to develop a trial like this. However, what happens is that this design finds the right dose, meaning the highest safe dose, in only 30 to 40% of the cases. And actually, with this design, it tends to identify toxic doses as being the right ones, wrongfully so. And that's another interesting thing about pharma, where actually it doesn't really matter much. You just enter your design. You know that your patients will suffer significant adverse events, but it is considered to be, unfortunately, part of the process. And one thing that I'm still appalled by working in pharma is how we simply accept that we're going to get it right only less than half of the time, to not say a third of the time. But of course, there are initiatives where, for example, the FDA has an initiative called Project Optimus, which was launched in January 2025, so just over a year ago, and whose purpose is to change these methodologies into methodologies that allow us to do dose optimization, where we prove with a lot more certainty that a dose is efficacious or not. Safe and efficacious or not, with a lot more accuracy. However, this is still not the mainstream in pharma, partly because there are not a lot of incentives to make this better, usually because these methodologies involved recruiting more patients, making larger trials, which means more time, harder to recruit, and all of that impacts into the speed and ROI aspects of what the pharmaceutical companies want to see. All right, so after our drug, out of the 10,000 drugs we started with, there was one drug that made it. We took that drug to a phase one trial. We showed that it is safe. And then we take that drug to a phase two. We selected a dose to test in phase two, which is the recommended phase two dose. And now, what do we want to do in a phase two trial? So I have personally, a very perhaps simplistic view of phase two trials, because people say, oh, it's super exciting. Now we're actually seeing if the drug works. Effectively, the way I see phase two trials is a way of collecting evidence before you go to the real deal, which is the phase three trial, where you actually need to prove that without a shadow of doubt that your drug actually works. And so this is why what we see here is we have these go, no-go decisions. So this is a phase where after we've shown that the drug is at least safe and does seem to have a little bit of efficacy, we would need to decide, are we taking it to a multi-million pound trial that will eventually lead to an approval? And knowing that if we fail, we would have spent a lot of money and a lot of resources and also treated a lot of people with the drug that doesn't work. And so this is the stage where everything is formulated as a no-go, a consider, or a go. By a go is we go to phase three. Consider is we might go to a phase three, or a no-go is we stop development of this drug, go back to the drawing board to 10,000 more compounds again. What I find interesting as well is that while in those questions that I was talking about, the classical trial and the Bayesian trial, and this is the case where people are a little bit more Bayesian because they are looking at the probability of an effect given a certain threshold. So we know that if we are, say, 80% sure that our drug is efficacious given a certain threshold, we will go forward. Effectively, the hard part of these trials is exactly that, is to know what is this good threshold that would then lead us to make a good decision in a phase three trial. And of course, I don't think this comes as a surprise. There are many aspects that weigh into this decision, whether it's efficacious, safe, whether what is the PK/PD data telling us, whether there's biomarker data that helps our case, and also what is the potential commercial fit. And even though this might seem that it's not important at this point, the reality is, and this is one of the challenges we oftentimes have working with different sponsors, is your drug works. There are people that are getting better or getting cured because of your drug, but just not enough. So say that the standard of care is that we see a response rate of 40%. And for example, I work a lot in oncology, and usually phase one, phase two trials are done as a third line treatment. So seeing response rates of 30, 40% is not unusual. But if the standard of care has a response rate of, say, 30%, and our drug seems to have a response rate of 35%, it is not a necessary go. And that depends on would we be able to commercialize the drug or not, and what are the competitors doing. And so there's having something with a 35 or a 40% response rate does not necessarily mean that the drug would even go to a phase three trial. So yeah, so essentially the main driver is to collect enough evidence to either kill the drug or progress to phase three. And this is uh, this is essentially based on how risk averse or not the sponsors are. It depends on money that they have available or not. All right, so before we go into phase three trials, I just wanted to quickly review something which people probably have seen loads and loads of times, but it's this difference between absolute and relative risk. And I promise this will have interesting clinical implications. So these are two examples where we have a control in a treated arm, and here again, here a control in a treated arm. And this is essentially in the control, let's assume that in the control people have an incidence of something, 50% of the people have an incidence of some disease, whereas in the treated arm, only 25%. So this could be cardiovascular disease, say myocardial infarction. So 50% in the control versus 25%. This means that the whatever treatment I'm giving them reduces their risk by 50%, right? In this case, however, what we see is we have a difference of 2% to 1%. And effectively, if I take that into account, this is my absolute risk, which is only 1% difference, whereas here it's the absolute risk is a 25% difference, but my relative risk is also 50%. And the devil lies in the details. And for example, a good example, practical example of this is the Jupiter trial, which at the time I was in uni studying medicine, and we did discuss this. And a few years later, when I was learning statistics, this was also used as a case study. What we see is, so the Jupiter trial is about rosuvastatin, so giving statins for primary prevention to people with essentially no risk factors or very little risk factors. And what you see is that the relative risk reduction reported by the trial is 44% in cardiovascular events, and this is a composite endpoint. However, and we think, oh wow, rosuvastatin is amazing because people have a reduction in 44% in their cardiovascular events. However, when we look at the absolute risk reduction, what we see is that it's actually a 0.6% per year in a reduction. So going from 1.36% to 0.77% with rosuvastatin. which then translates into this NNT, which is the number needed to treat to effectively prevent one event, would be 95 people. So I need to give rosuvastatin to 95 people over the course of 2 years in order to prevent one cardiovascular event. And of course, this poses the question, is it worth or not giving this drug to patients? This is not the topic of this discussion, so I'm not going to get into that. But it's just something to consider when we see these very large relative risk reductions, whether they are clinically impactful or not. And this brings us to phase three, because in phase three, we also have the hazard ratios, usually. So oftentimes in most of the trials, not all of them, but most of the trials, the regulators want time to event endpoints. So in cancer, for example, we see a lot of progression-free survival or overall survival, so PFS and OS, and we have hazard ratios. And hazard ratios essentially just tell us what is the difference in the probability of, let's say, dying for overall survival in the treated versus the placebo. And for example, here we see that in the drug zero, let's say drug zero is the placebo, there is roughly a 50% chance of dying at around 9 months, whereas our 50% chance of dying would be at around 28, 29 months for our drug. And this suggests that we have a very significant effect in our drug. In this particular case, yes, because our chances of dying go from, say, 9 months to 29 months. So we're sort of triplicating the survival odds. However, this is, even though in this particular case this is great, what in reality happens is a trial is designed such that we consider a potential hazard ratio for improvement. So when I say I'm going to recruit 300 patients because I'm expecting to see a hazard ratio of 0.5 or 0.75, in order to prove my result, what happens is if I go for a hazard ratio of 0.5, and 0.5 means that, for example, if my median overall survival in the control arm is 10 months, my median OS in my treated arm would be 20 months, which is great. So it means I'm essentially doubling the survival time from 10 months to, so just under a year to almost 2 years of survival, of median overall survival in my treated group. So essentially a good result. But at the same time, my median OS, if my median OS is 2 months in my control arm, so usually people that have this diagnosis, and this happens in loads of cancers, have a 2-month expected survival, and I have a hazard ratio of 0.5 as well, my median OS is now 4 months, which means that my benefit is increasing lifespan on average by 2 months. And this is something that I always think about a lot. And trust me, I've designed quite a few trials like this where I can see that from a maths perspective, this is all correct. But from a clinical perspective, this is probably not very meaningful. But then again, it depends on what we consider to be actually meaningful or not. But the same numbers go behind the two of these, and the same numbers will go behind this drug getting an approval. In effect, effectively, there have been many approved drugs that are basically buying people 3 months, 2 months, 3 months, 4 months on as an average effect. So yeah, so this was sort of a quick rundown on some aspects, a bit mathematical, of clinical development and in some issues or caveats that exist between translating what the data shows and then actually giving that to patients and seeing that work in real life. So yeah, my contact details are over there. I'm usually
Yes.
active on LinkedIn. Feel free to connect. And yeah, Neil, thank you so much for organizing.
No, thank you, Miguel. Loving the final slide and all the slides. So the antlers with yeah, I like that. Very, very humorous. So like Miguel said, reach out to him via his email and LinkedIn, and we'll be placing that once we release the recording on our YouTube channel. I have questions, Miguel.
All right.
As you
Happy to
know, as you
answer.
know, the pharmaceutical clinician hub, but I'm using the word omphalos to mix it up. We're slowly building, and the reason is very simple, that there is a clear disconnect between people in medicine and the pharmaceutical industry. I think in the sort of shared understanding and real sort of life applications. You are unique, which is really good because you have that medical and pharma background. So. Um, I'm going to kick off with a few questions, and it's probably because of my lack of understanding and experience, and it's in no particular order. So there was a slide which I really liked. I'm going to guess, was it an anti-aging drug? I mean, you showed two individuals side by side. It was a an elderly chap and
Oh
then
yeah, yeah. I can go back to that quickly because
um
I do
and
like the guy that's like, I'm winning.
yeah, yeah. No, it was interesting. I was like, oh, what kind of drug was that? But I'm just playing it. But yes, so for the first time, having been involved in clinical trials for so many years, is I actually now see what you're experiencing on the other side, but you have obviously the two hats, you've got the clinical perspective and the data perspective, whereas obviously I think the vast majority of clinicians who are not sort of dual qualified, they just see the patient side. And that is, I think this is an interesting problem to be solved. I mean, I can imagine your frustrations, like you said, like because you have that insight and the expertise, you can then visualize, okay, I'm seeing um I need to actually put my reading glasses on to just remember. Yeah, so I'm seeing
Yeah,
like headaches,
bunch of stuff.
yeah, just quickly put it into my head. And then you're able to imagine, okay, well, the patient is sort of presenting this way. But the vast majority of people in pharma don't have your skill set, right? So they'll just see the numbers, I guess, and the grading. Or have I misunderstood that side? Is it only clinicians that have the dual hats that then get the data? But I don't
Yeah,
think
no,
so.
no. I mean, lots of people end up seeing, well, this is mock data, so it's not real patient data.
Sure,
But
yeah.
a lot of
It's
people,
definitely important for us to say that, yeah, no,
yeah, a
perfect.
lot of people, so a lot of statisticians who have absolutely no clinical training, data managers who also don't necessarily
Mm,
have clinical training,
see
will
that,
see
yeah.
this data. And one interesting example I have was in a trial that I was running in, not only running, but I was also part of the safety committee. One patient died,
Mm, okay.
and we the first thing that the sponsor did was get the data and look at the data to see what had happened.
Mm,
And so the patient had died, and the adverse event that had been reported was vomiting.
okay.
And so you have vomiting, probably grade three, or actually ended up being grade five. And it's like, okay, so how does someone die from vomiting?
Yes,
Right?
yes, yes, of course.
In effectively when then the,
Electrolyte disturbances, maybe. I don't know.
but yeah, but you don't know, you have no idea. You
No,
just
no,
see
yeah.
vomiting that started probably just roughly 24 hours before death. But this information was also not something you could see from the data.
Mm,
And someone died of vomiting. How does that happen? And
mm,
this is a patient with cancer, so someone who's very ill with metastatic cancer. So we had a meeting to discuss the case, and the medical director had spoken with the investigator that was in charge of that site, of the site where the patient was going. Turns out that the grade five vomiting was someone who had their treatment a couple days before they died. They had gone to a restaurant after that. They fell ill, and the husband of the
mm.
patient thought that he was
Gastroenteritis.
just gastroenteritis. She didn't. She decided not to go back to the hospital because she didn't
Massive.
feel like that was something that was due
Mm,
to her treatment. She stayed
yeah.
home, and she ended up dying. She ended up being found in her bathroom, uh, just lying on the floor dead. And this
Oh,
is what a grade 5 vomiting episode looks like. But
mm.
I can imagine 100 scenarios from seeing a row saying grade 5 vomiting. Yeah.
That is interesting. And it's also, if you deconstruct everything that you said, I mean, the thing that I've experienced in the clinical trial space is we always have to decide, is it related to the drug? Is it not? Is there sort of shades of gray? And then you consent, particularly in the cancer space, you're consenting patients to anything that could potentially go wrong with the drug. And you just wonder, like, yeah, I'm just hypothesizing on the spot here. Yeah, we consent to the drug, and nausea, vomiting, certainly is a side effect of all drugs. But do then patients maybe misunderstand that they may think, oh, this isn't drug related, so I'm not going to report it. But maybe we should say, if you just feel unwell for whatever reason, well, I think that's on there actually. Yeah. I don't know. I'm just thinking on the spot.
But there is
Interesting.
that's actually a good point because in general, so what happens is you have AEs that the patient just reports because they felt they vomited, so that's something that
Mm.
they report.
Yep.
But then you also have elicited AEs, which is adverse events that you ask that people might not necessarily be aware of. And actually, that's very common in vaccines because in vaccines you're just supposed to give a shot, and people are not supposed to feel anything else, right?
Yes,
And
yes.
people just go about their lives as normal. But then you go and ask them, oh, actually, did you feel any pain in your arm? Yeah, of course you do.
Mm.
You usually do. What was, then you try to grade that pain. And so there's this mix of adverse events that the patients will just go and report because they know they're in a clinical trial and they might know which adverse events they can expect, and then others that you need to elicit from them. But all of that biases the
Yes.
clinical presentation, or it creates a difference between the clinical presentation and what you actually have reported.
Ah, definitely. Very interesting discussion. Also, again, lack of knowledge, hence why we are closing the gap. Um, the no-go aspect. Now, this interests me. So, I've never really been involved in that in the uh, pharmaceutical space, but I think this no-go concept does happen when it comes to the funding space. I remember this general sort of theme, should we fund this or should I don't know, kind of thing. I mean, I don't apply for grants anymore. Long,
Yeah.
long past that. But right. So do you ever get into a situation where you have people around the table, you got these committees, like you say, and some say go, some say no go. Like, what do you do? Does it then just go into consider? Is that how it works? Or
Yeah, so it's a tough.
tough one? Yeah, because
So
that
this
that might
actually
be me in those
happens.
meetings.
This actually happens in two stages. One is
Yes.
um, the guessing stage where you have not run your trial, you're planning your trial, and you are trying to decide what would good look like, what would so-so look like, and what is a complete no uh, from the get-go. So that
Yeah.
has impact on how you design your trial. And then after you design your trial, you actually do the trial. Then you have these discussions where you say, well,
Mm.
uh, because it's how you see the glass, half full or half empty.
Exactly.
Because, for example, we had an example of a melanoma trial uh, in
Okay.
which you could see some patients actually showing some complete responses, some partial responses in some cases. And
Mm.
this is metastatic melanoma, so relatively difficult to treat. Um,
Yes.
and and you're like, well, actually, some people have complete responses due to this treatment.
Hmm.
However, when you look at the overall response rate, it's really low and it's uh, comparable to standard of care.
Hmm.
And this is, and then you say, well, I know this, my drug is actually helping people that had no other options.
Hmm.
Uh, but at the same time, I'm not beating the response rate in the standard of care. In this
Understood. Yeah.
particular case, also partly, as you mentioned, the money aspect of things, partly due to funding, partly because they couldn't identify a subgroup of patients using, for example, a biomarker that they could say, well, actually, if we just get this subgroup of people, we can take this forward in the
Hmm.
in the program with Skilled.
Interesting. Yeah. Well, one day, if the stars aligned, I'll get to sneak into one of those meetings. Um, and yes, this is again a deficiency of mine. I claim I've got trial experience, but clearly, I'm not doing it properly. Um, Obviously, patient safety I am, but my general knowledge
Well,
is
the thing, I'm pretty sure you're doing it properly because you are treating the patients.
yes, we follow the protocols. Right at the start, you talked about, and this is the fascinating space that I've kind of got caught out when it comes to just talking to patients about novel therapies. So I want to get better, and I think I'm not the only clinician. I want to move away from saying, oh, we've got a novel therapy and it works to reduce the cancer and it works in, because the science of these protocols can go on for many pages, but in a
Yeah.
clinical consultation, you don't have the time. And I think clinicians really push on about, what are the side effects? What are we looking out for if things go wrong? Like reactions on the first day of treatment, so on and so forth, and when the CT scan will be happening if it's a cancer drug, etc. But this aspect doesn't get taught very well in the medical schools and in general medical training, I think, is, the concepts of all that bit that you talked about. So it is the screening, the hits, the targets, the validation. I mean,
Mhm.
they're all cool words, and it's kind of just
Yeah.
trying to just get that in a language that I think yeah, no, I just think that area is quite interesting to focus in on because I say this, I'll try and connect why I'm thinking this way as real life experience. So there is a fragmentation there from what I've seen and the observation when you talk to patients about trials and, you know, they're very respectful and so they nod and go, yeah, but you can tell that they're like, oh, I'd like to know
Yeah, yeah, what's good?
the, the
What
real
are you
science.
giving?
Where's this magic juice come from, kind of thing? But the clinicians like, we'll give it to you, and the nurses will, put it up there. We'll be
Yeah.
around. So yeah, I mean, yeah, it's like also figuring out how do we get better in this aspect going forward, and is that an
Mhm,
area that we can really zone in on for clinicians who are sort of across the two spaces? How can pharmaceutical individuals like yourself, but obviously you've got the two hats, so it doesn't apply fully because you have that skill set. But
mhm.
something really that I think, yeah, maybe it's more of a statement. Yeah, I'm iterating live, so I don't know. But
No,
do
so
you,
I
do
think
you
it's
see that's a problem? Do you see that it's an assumed thing that people in pharma say, well, yeah, I'm sure the clinicians know what's going on there, but
yeah.
maybe they
Well,
don't.
because what I think so this space and yes, actually this CMC, which is the chemistry manufacturing part in the
Yeah,
GLP talks, etc.
that's
All
it.
of this is very, very heavily regulated, and we know that farming generally is very regulated. But the when you get to the stage that you have a drug to give to an actual human being,
Mm,
there has been there have been a lot of checks done, not just necessarily by the pharmaceutical company. So there's things called CDMOs, which are contract manufacturing organizations. So some
mm,
whose goal is to help pharmaceutical companies going from here to here by helping them with the lab processes and purifying the drugs and then making the drug into
yes,
something that is produced consistently across all the batches that are going to be given to people.
that's it. Yeah.
And there is a lot that goes into that. And then not just that into that, but also in then how figuring out how you can transport these things so that you can run your trial globally because the pharmaceutical industry is completely global.
Yes.
Um, in essentially what happens is that you end up having one lab or a few labs that are manufacturing these products. By the time they are effectively approved, this is also actually checked by places like the MHRA. The MHRA, for example, has labs that will check batches of things coming into the UK to see
Mm,
if they are actually doing what they're supposed to do before they are administered to people. So effectively, from a regulation perspective, and if having tight protocols in place, this works actually really, really well. So by the time,
Yes.
so
Yeah.
basically the way your drug is going to look like when it is approved is what it looks like when it's being tested from first patient in phase one.
Hmm. No, I can imagine the regulation is solid. It's just, yeah, maybe I'm just trying to think, would clinicians benefit from seeing that side?
There
Um,
is, so I know, for example, in some places like in Stevenage, there's the bioscience catalyst. There's a facility there for the production of um, cell therapies. So effectively,
okay,
examples
interesting.
have well, multiple labs, which are essentially their own closed units.
Hmm.
And so for cell therapy, that's actually really, really interesting because with cell therapy, usually it's autologous. So you take cells out of your patient, which could be, say, in the US. It gets shipped over to this site in Stevenage. It's called Bioscience Catalyst. It's called Catapult. It goes to this lab in Stevenage. It gets processed there in sort of like a one-way system across the lab, which is now
Hmm.
being partly automated by robots that can just, take something from
Cool.
this
Game
incubator,
changing,
go to
obviously.
the next site, put it on a shell or something like that. Um, in following this protocol, and then at the end of this, you have your cells, say CAR-T cells, ready to go
Okay,
back to
yeah.
the US to be given back to the patient. Um, so yeah, so this is, and of course, this is the example of cell therapy, but the same would applies
Same
to,
applies.
say, small molecule drugs.
Cool. I'll make a visit to Stevenage then one day.
Yeah, it's
Well,
called Catapult. Yeah, it's pretty
oh,
cool.
thank you. Well, thanks so much, Miguel. This was fantastic. I learned so much, and I'm sure, all our followers of the Pharmaceutical Clinician Hub will really benefit from your talk today. And as we continue to build, I think the journey is going to be very exciting. So very happy that you can join us on board our journey.
Thanks so
Okay,
much, Nick.
take care.
It's a pleasure.
Have
Cheers.
a good
Take care.
evening. Bye bye.
Bye bye.